This video will continue our study of macromolecules to review features of carbohydrates. From the previous video, you may recall that carbohydrates are important for cellular energy and give structure to some cells. This video lesson will help us to meet our learning objective to describe and identify the four major biological macromolecules. Carbohydrates are also known as polysaccharides and polymers made of repeating subunits called monosaccharides. Each monosaccharide has a molecular formula of CH2ON, where N represents the factor that each can be multiplied by. This factor ranges from 3 to 7. Additionally, we will see that the monosaccharides contain carbon atoms with more than one hydroxyl group. Monosaccharides are primarily classified based upon the total number of carbon atoms. For example, a monosaccharide with 3 carbon atoms is a triose, 4 a tetrose, 5 a pentose, and 6 a hexose. Monosaccharides are also classified based upon the location of a carbonyl group. If the carbonyl group is at the end, it's an aldose. If it's internal to the carbon skeleton, it's a ketose. Figure 212 from the textbook demonstrates this carbonyl location in a sample ketose and aldose. Panels A and B show the carbonyl groups highlighted in yellow in each fructose and glucose. Figure 212 also shows the various ways to represent monosaccharide structure. We can look at them in the straight chain form, such as in panel A and B, the ring formation, Haworth projection, chair form, and ball and stick form. One can count the carbon atoms by finding the carbonyl group. The first carbon nearest it, or in it, is carbon 1, then count in sequence. Monosaccharides do not tend to exist in the straight chain form in a living environment. Instead, it takes on a closed ring shape. This shape will have a significant impact upon the function of the monosaccharide. To count the carbon atoms in the ring formation, find the carbon that makes up the carbonyl group, which will be carbon 1 or 2. In this example of deoxyribose, an aldose, carbon 1 is on the far right-hand side. Count counterclockwise and don't forget the projection. We can do the same for a ketose. The carbonyl is on carbon 2. So in the ring formation, carbon 1 is projecting off to the right in this image. Carbon 2 is the carbonyl group, and then continue to count counterclockwise. Once in the ring formation, monosaccharides can form isomers. To determine whether a molecule is in the L, left, or D, right form, look at the hydroxyl group second furthest from C1. If it's to the left, it's in the L form. If it's to the right, it's in the D form. Another structural variation is the alpha versus beta form. This is related to the hydroxyl group of carbon 1. When in the ring form, if the C1 hydroxyl group projects downward, it's classified as the alpha form. If the C1 hydroxyl group projects upward, it's classified as beta form. In summary, polysaccharides are polymers of monosaccharides. Monosaccharides are made of carbon, hydrogen, and oxygen. Monosaccharides are classified based upon the number of carbon atoms and location of carbonyl groups, and common isomers exist in the alpha or beta form.